Okay. Day 15. It was a tough day, so I'm probably gonna be slow, but we'll see. And we're gonna start from a sidetrack. Remember this? We started the project building. For some reason it's still spinning. All done. So I started a local host and check this out. Cognitive Lycon. Human brain, a dog, civilization, ant colony. I mean, obviously the numbers are totally off. The shapes are off as well. But this is a start. Look at this. They should be in the same axis. AI chatbot. It's flat. It has no future or past. Look at this. That's so funny. Beehive. Oh, that's a linear. So AGI, family, company. What is this? Honeybee? 30 days? They're thinking 30 days. Honeybee and beehive. In the future, oh, here you go. API error 500. Anyways, that was a good experiment. I'll answer a couple of questions today, and probably that would be it. Okay, so that's it. Yeah, that's the question that I try to ask. I don't know. Yeah, what is plasmid? Ah, I remember that is extra DNA chunk. Is a small circular loop of DNA that exists inside bacteria, separate from the main chromosome. And then we run into quick sidetrack, not a quick, but big one, like what is DNA actually made from? And then we went into retroviruses information and then receptors and how was that? Enhancers and receptors. Enhancers, that's a chunk inside of DNA with information, how receptors should be built, and receptors, that's the one. Think of the bacteria chromosome as the main hard drive and the plasmid as a USB stick. It's extra, optional, independent DNA that replicates on its own. That's crazy part. It replicates on its own. What's on a plasmid? Usually, what's on? Just a few genes, often genes that give bacterium some survival advantage. The most important example, antibiotics resistance genes. Whoa. Well, that's where it's saved. A bacterium might carry a plasmid with genes that produce an enzyme that destroy penicillin. That's crazy. That's so crazy. We evolved new ways to kill bacteria, and they evolve plasmid to produce enzyme to kill our medicine.
Plasmid.
That's pretty cool.
Plasmid.
I mean, not cool for people, but cool for bacteria. Without that plasmid, it dies when you take antibiotics. With it, it survives. The scary part, bacteria can share plasmid with each other. One bacterium can pass a copy of its plasmid to a neighboring bacteria through a process called conjugate.
Conjugation.
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 Conjugation. Conjugation.
Conjugation.
Conjugation.
Conjugation.
Essentially, horizontal gene transfer. This means antibiotics resistance can spread between bacteria that aren't even the same species. Oh, that's crazy. So they develop how to protect it and give it to different bacteria, not their own species. That's super dangerous. The resistant bacterium in your gut can handle the resistant gene to a completely different species sitting next to it. This is a major reason antibiotic resistance is spreading so fast globally. It's not just evolution through mutation, it's bacteria literally mailing each other cheat codes. Wow, why scientists love plasmids? Plasmids are one of the most important tools in biotechnology because they are nature's ready-made DNA delivery vehicle. Scientists use them too. Nature's ready-made DNA delivery vehicle. Scientists clone genes. Take a human gene, say the insulin gene, insert it into the plasmid, put that plasmid into the bacteria. The bacteria now reads the gene and produce human insulin. I mean, yeah, but you can transfer them into bacteria only. How can you transfer them into human? Probably not. Cannot. This is exactly how most insulin for diabetes has been made since 90s. Bacteria carrying plasmids with the human insulin gene grown in giant fermentation tanks. Deliver genes into cells for research. Want to study what a gene does? Put it into plasmid, insert plasmid into cell, watch what happens. But can you insert it in a human cell? Can you insert it into human cell? Or only bacteria? (56.2 45) CRISP delivery. The guide RNA and Cas9 instructions are often delivered into cells via plasmid. So yeah, I ask a stupid question because right now I can see it. Programming knowledge of a plugin or package that can be installed into any compatible host. Both, but they behave differently. In bacteria, plasmids replicates independently and persist indefinitely. The bacterium treats it as a natural part of its biology. It copies the plasmids every time it divides, so all daughters cell inherit it. This is why bacteria are the workhorse for mass production proteins like insulin. You set up once, the bacteria keep capping themselves with plasmid forever. In human cells, you can absolutely get plasmids in. The process is called transfection. Common methods includes electroporation, brief electric pulse, punches temporary holes in the membrane, liquefaction, wrapping the plasmid in a lipid bubble that fuses with the membrane, or viral vectors using delivery vehicle hijacking the virus' natural ability to inject generic material. The key difference: human cells generally don't replicate plasmids. The plasmid gets in the cell, reads it, and makes the protein you wanted. But when the cell divides, the plasmid doesn't copy itself. It ends up in one of the daughter cell or get degraded. So the effect is temporary. Guard over days, weeks. If you want permanent expression in human cells, you have three main options: viral integration, CRISP, episomal vectors. I don't know what's that, but that's interesting. Okay, this is number 4. I don't know. Krebs cycle, also called the citric acid cycle. I don't know. I'm not even gonna guess. No worries, this one connects directly to things you already know. Remember the energy story? Your cell break down glucose to make ATP. Yes, there is 2 different breakdown mechanisms. Aerobic and anaerobic respiration. I remember that happens 3 stages, and the Krebs cycle is the middle one. Stage 1, glucolysis happens in the cytoplasm. Glucolysis happens in cytoplasm outside mitochondria. Glucose, 6-carbon, is split into 2 molecules of pyruvate, 3 carbons each. Produce a small amount of ATP, net 2. Doesn't require oxygen. Yep, this anaerobic part. I just called it. Stage 1. Stage 2. Krebs cycle happens inside the mitochondria. Pyruvate enters the mitochondria, gets converted into 2-carbon molecule, and feeds into circular chain of chemical reaction. Each turn of the cycle strips of carbon atoms, release CO2. This is where the CO2 you exhale comes from, and harvest high energy electrons, loading them into carrier molecules NADH, and FADH2 produces only 2 ATP directly by the, and then electron transport chain happens on the inner mitochondria membrane. Electron transport chain. The electron carries from the Krebs cycles dump the electrons into chain protein. That's above my head. For 55% exactly. 4 left. Let's go. What does vitro versus vivo means? Oh my god, I'm totally losing it. I don't know. I'm gonna drop from 55 back to, I don't know, in vitro, in glass, experiments done outside in living organism, in the test tube, petri dish, or cell culture. When researchers say we show this work in vitro, they mean it work in dish. Oh, that's cool. We showed this works in vitro. In vitro, in vivo, in the living, experiments done inside a living organism, in a mouse or rat, humans. We show this work in vivo. I need to know how it's pronounced.
Vitro. Vivo.
Okay, exactly how I thought. Why the distinction matter enormously? Thousands of cancer drugs work in vitro. You can kill cancer cells in dish with a handgun. The question is always whether it works in vivo, inside the real body, with immune system, blood flow, organ interactions, drug metabolism, and thousand other variables that dish doesn't have. The pipeline for any drugs of therapy goes in vitro, in vivo, clinical trials, human. Interesting. The vast majority of things that works in step 1 fail in step 2. Most things that works in step 2 fails in step 3. This is why drugs development takes 10 plus years and cost billions. The third term, you encounter ex vivo, out of the living. Take cell out of a patient, modify them in lab, put them back, creates therapy for sickle cell. No, 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 no. 53. Oh, I dropped 1.2. Wow. What is the protein shape and why does it matter? So I think that's where alpha folds play very big important role. The protein, since it exists from amino acids, there is many variations of protein. So many variations how you mix those amino acids together. But what's interesting about that protein can fold in different shapes, and those shapes and folds really determine of its function, and it also can carry different charge. A good example also would be in antibodies. Antibodies, it's a protein that's shaped specifically for the pathogens to match pathogens. Another good example would be the receptors on a cell membrane. They call receivers, I believe. Receptors. Yeah, they call receptors. Sorry. So the shapes is matter because it can grab different and attach to the different counterparts. And in a lot of cases, it's produced kind of a lock key combination. Where the protein plays the lock role. Maybe sometimes it's play key role as well. Let's see. You connect and can say across the whole quiz. Proteins are made of amino acids, correct? And you said it with confidence this time. Progress. Thank you. Progress. Many variations in how you mix amino acids, correct? 20 amino acids types, chain of hundreds of thousands long, astronomical numbers of possible sequences. Chains of hundreds to thousands long. Wow. Proteins fold into shapes that determine function, correct? This is the central principle of protein biology. Can create different charges, correct? The folded surface has patches of positive and negative, neutral and neutral charge, plus hydrophilic regions. This charge landscape determines what the protein can interact with. Antibodies shapes to match pathogens. Correct. Each antibody has a unique binding site shapes to grab one specific antigen. Your immune system generates millions of different antibody shapes to cover millions of possible threats. Membrane receptors, correct. Protein embedded in the membrane with a specific shape on the outside that binds specific signal molecules. Lock and key, correct. Damn. Proteins can be either the lock or the key depending on the context. Enzyme as a lock, active sites, and substance as a key. Hormones as a key, and the receptors as a lock. AlphaFold, correct. Before AlphaFold 2020, determining a single protein shapes took months to years using X-rays crystallography. AlphaFolds predict shapes in minutes and has now predicted structure for essentially every known protein, 200 millions. Wow, it's arguably the biggest single contribution of AI to science so far. The 4 levels of structure you didn't mention. Primary, the sequence of amino acids in a chain, just the order, like the letters in the sentence. The second, local folding patterns. Amino acids chain naturally twist into alpha helixes, like a spiral staircases, or flatten into beta sheets, like a pleated fan. These are driven by hydrogen bonds between nearby amino acids. Sequence of amino acids in the chain. So sequence is important, the folding pattern is important. Tier theory, the overall 3D shapes of the entire chain into compact globular structure. This is what AlphaFold predicts and what determines the function. Quaternary. Some proteins are made of multiple chains assembled together. Hemoglobin is 4 separate chains, 2 alpha, 2 beta, that combine into one function unit. Not all proteins have this level. Why misfolding is there? If protein folds wrong, it doesn't just stop working, it can become toxin. Prion diseases, mad cow diseases, are caused by a single misfold protein that acts as a template, causing normal copies of the same protein to misfold in a chain reaction. Alzheimer and Parkinson also involve accumulation of misfolded protein. The shape isn't just important, it's getting it wrong can kill you. Wow. Second to last. Let's push it. Ooh. What is the difference between benign tumor and the malignant tumor? Wow, I don't know. Honestly, I don't know. I know the tumor is abnormal growth of the tissue. So in this particular way, tumor is when cell disconnected from the signals of neighboring cell, and it's now think it's by its own kind of fallen back into the amoeba behavior, and what it does is just divide and grows, and that's what we call tumor. And benign and malignant tumor, probably a bad one and good one in the biological terms. That's my guess. While it's thinking, I'm gonna listen the proper pronunciation of that.
benign,
Benign, benign, benign.
malignant,
Malignant. You guess is right. And quick note, I think you mean amoeba behavior, not amygdala. Amygdala is the brain region involved in fear emotion. Call to your excellent analogy from Call back to your excellent analogy from question 14. Tumor abnormal growth cell disconnects from signals. Correct being and malignant, good and bad. Correct in spirit. Here's the actual difference. Benign tumor grows slowly. So this stuff, right? Like wrinkles? No, not the wrinkles. What's the name of it? Like this stuff, right? That's benign. Stay contained, surrounded by a capsule, doesn't invade neighboring tissues, does not spread to other parts of the body, usually not life-threatening unless it's persistent on something important. A benign brain tumor can still kill you by compressing brain tissue. Mhm. Cells still look relatively normal under microscope. Example, moles. That's what I was looking, moles. Lipomas, fat limbs under the skin, uterine fibroids. I have no idea what it is. Malig- malignant tumor, cancer. Pretty much it's cancer. Grows aggressively, invades surrounding tissue, pushes into the destroying neighbors. No capsule, no boundaries. Metastasis, this is the key word. Malign cell breaks off, enters the bloodstream or lymphatic system, travels to distant organ and start growing new tumors there. Lung cancer spreading to the brain, breast cancer spreading to bone. This is what makes cancer lethal in most cases. Not the original tumor, but the metastasis. Cell looks abnormal. Under triggers the critical distinction in one sentence. Mid tumor stay put, malignant tumor spreads. What? Correct on what tumor is and the good and bad framing, but didn't know the actual distinction, invasion and metastasis. Okay, the last one. It took 15 days to answer 50 questions. Why do we age? Oh, that's a good one. That's a good question. So, yeah, age is very interesting. So, accumulation of bad mutations, accumulation of misfolded proteins, uh, bad tissues, accumulation of bad stuff. But what's interesting is that age was kind of given and accepted. But now, latest development and science show that it's actually a gene for the age, and age could be treated as disease, not inevitable. What's that mean? That I forgot his name, Michael someone, not 11, but Michael, maybe not a Michael name, but there is a big the scientist that's pushing it very hard, that agenda. They already know what gene actually turn off aging and roll back to regeneration, and you can roll back clock not to the when you baby, but to the certain age, and then it stopped. And it's actually in some organism they live forever. I forget the organism name, but they live forever. Some yeah, I don't remember them. And now that scientists already, I believe, in they prove it in vivo. So they prove it in vitro first, then in vivo in mice. They were able to regenerate the age completely, and now they're doing human trials. What cool about AI is that AI helps them to found the proper proteins and shapes of those proteins who can deliver that signals to turn those genes on to roll back your age. Clips. Great final answer. You are mixing some real cutting edge science with some oversimplification. Let me. Yeah. Oversimplification. Accumulation of bad mutation with false protein damage tissue corrects. This is the wear and tear damage accumulation theory of aging, and it's one of the real mechanism. Aging could be treated as disease. This real and increasingly mainstream position. The scientist you are thinking of is almost certainly David Sinclair. Yes, for sure, that's him, who has been the loudest advocate for reframing aging as a treatable condition. He wrote Lifespan, Why We Age and Why We Don't Have to. Genes that roll back the clock. You describing Yamanaka's factors. OCT4, SOX2, Clifford, and yeah. Yama won the Nobel Prize in 2012 for discovering that these four genes can reprogram adult cells back into stem cells, in, induced pluripotent stem cells. Sinclair's lab and others have shown that partial reprogramming, turning into these factors briefly, not fully, can reverse age-related changes in cells and tissue without turning them all the way back to the stem cells. Proven in mice corrects. Sinclair's lab published the results showing particular epigenetic reprogramming restored vision on old mice by resetting the epigenetic marks on retinal cells to younger state. Other groups have shown lifespan extension and tissue rejuvenation in mice using similar approaches. Wow. Regeneration is age completely and doing human trials. This other state where we are, the mouse result showed partial rejuvenation of specific tissue, not complete age reversal. Human trial for reprogramming based therapies are in very early stages. We are far from rolling back your age in humans. I don't know. Organisms that live forever. You're thinking of Turritopsis dolphin, the immortal jellyfish. Yes, when stressed or aging, it can revert its cells back to younger state and restart its life cycle, especially biological reprogramming in nature. There is also the Hydra, which shows negligible senescence, and the naked mole rat, which barely age and almost never get cancer despite living 30 plus years. Extraordinary for rodent. AI helped find proteins in shape. Correct. AlphaFold related tools. But it's not AlphaFold. Like they have their own models. The Sinclair lab. The full picture of YVH is not one thing. There are actually several mechanisms running simultaneously, and biologists have organized them into hallmarks of aging. A landmark 2013 paper updated in 2023. The main one. Telomere shortening. Oh yes, that's the ending of the DNA or chromosome. And when they shorten, when the cells divide too many times, basically 12 or something like this, then it's basically destroyed molecular structure. Example would be the shoelaces. Chromosome caps erode with each division. Call back to question number 3. High flick limit. Epigenetic drift. The epigenetic marks that define the cell identity gradually become noisy and disordered. Sinclair calls this information theory of aging. Information theory of aging. It's not that the DNA is damaged, it's that the epigenetic instruction for reading the DNA get corrupted like a scratch CD. The data is still there, but the reading errors accumulate. Mitochondrial dysfunction. Mitochondria become less efficient with age. Call back to Q3. Cellular senescence. All cells stop dividing but don't die. Oh, interesting. They sit there secreting inflammatory signals that damage neighboring tissue. These zombie cells accumulate with age. Drug called senolysis that clear senescent cells have shown rejuvenation effect in mice. Oh, interesting. I need to look up into this. Stem cells exhaustion. Your regenerative capacity declines as stem cells pools shrinks. I have no idea we have a stem cells pool. Accumulated protein damage, misfolded protein cell garbage. You mentioned this. Chronic inflammation, low grade body-wide inflammation increase with age inflaming. That's what I have. Like I got the bites and like look at this. It's not the bites. I even like touch some plant and now it's all over my hand. The exciting part of current research is that many of these hallmarks appear to be interconnected through epigenetics, which suggests that resetting the epigenome, what Sinclair is pursuing, might address multiple hallmarks simultaneously rather than needing separate interventions. 54. Wow. 54%, 27 out of 50, final score. Want me to generate the full gap analysis? Yes. It took 15 days, half a month, probably 20 with breaks. That was crazy. But I loved it. Oh, thank you for the comment. Yeah. We will keep going. Appreciate support. So now when we have a gap analysis, we can identify what we need to fix and then jump into the catching up with the latest and greatest development and see where we can apply Fox mentality. Like in math, you have two types of mentality. Fox, that's kind of jumping in the concepts, and then hedgehog, that's dive deep in certain area. So we're gonna fox around. And by the way, did you see the Likeon that we created last time? The Cognitive Likeon website? Start the time. If you're still here, I'll show you. Was it port 80? API coverage. Probably need a new session. Step. I just downloaded that ID, so not sure how to use it properly. Right here, can I kill it? It's still saying it's running. The gap analysis is here. Let's take a look. Whoa, that's a big one. Did it start? That's a back end with the fiber. Where is the front end? Why it's still running? That's so unexpected. Come on, start the front end. Backend is here running. Front end is not running. Yeah, I thought to show you a cognitive icon that we built it. 3D representation. So gap analysis of my knowledge, May 5th, Score 27, 54%. Open questions, that's important. Open questions, no multiple choice. No Googling. Yeah, I think like multiple choice, first of all, it always gives you a clue, at least a little bit, but you can exclude bad answers, and also it's kind of limits you in a way. Prokaryotes versus eukaryotes. This one I know already. Do I have like a 1 for something? Mutation, 0.7. Cancer, I really know cancer. Photosynthesis, then enzyme. Natural selection, look at this. Mitochondria, I get 1 point. Damn. That's what's up. Ecosystem. Okay. Performs by demand. Strong, 70+%. Are we running or not? No, we're still not running. You understand this conceptually. Genetic and evolution. Natural selection, mutation, epigenetic, Krebs, aging. You have strong intuition. Ecology, 90%. Microbiome. System level thinking is your strongest domain. That's what's up. The system level thinking is the strongest domain. That's what we think about. We're thinking about system, how to build them, how to understand them, how to use them, about interconnections, feedback, unintended consequences, and emerging behavior naturally. Moderate. Partial knowledge gaps in scientific. Energy metabolism, yes. Psychology and system. Psychology? Why is psychology? Okay. Microbiology, pathology. Ribosome 1. Molecular biology. 50%, not bad. Weak. Major gaps. Cell biology. Yeah. Stem cells. Mitosis, meiosis, osmosis, somatic germ cells. Yeah. Terminology 0. That's funny. Terminology 0. So now. Recurrent pattern. Strong scientific vocabulary. The enzyme protein default. System thinking over reductionism. Frontier knowledge boundary. The map. How big is biology? Do I know 10% or 0.01%? Wow, you asked that at start. That's a very good question to ask. Five dash 8% of undergraduate biology education, but distributed unusually. That's a good. The good frame, right? Personalized study map. Plug the critical gaps. Week 1, 3, the foundation everything else built on. The 4 biomolecules. carbohydrates, lipids, proteins, nucleic acids. Know what each made of. Cell division, mitosis, meiosis, why they exist. NAD plus metabolism. Retake this quiz in 2 weeks to measure improvements. Same format, different questions, same scoring. Luck in the 4 biomolecules and the nucleotides amino acids distinction today. I thinking I took it in 1 day. Baseline established day 1, score 54. Track from now there. We're gonna do report. I finish. I finished. Um, 5 of them I didn't know at all. Can I do enter? I don't know how it's calculated, but typical beginner would score around 70 on cell biology basics and 10% of CRISP aging microbiome. Use the inverse. That's what's up. Your knowledge is shaped like a dumbbell. Strong at the what, meta, and why level. Strong at the cutting edge application level. Hollow in the middle where the textbook fundamental lives. Let's go. That was fun. See you tomorrow.